So the aim of the today's video is to understand and interpret the phylogenetic tree. In the previous video, we learned to construct a phylogenetic tree using Mega 11 software. If you have missed that video, I recommend you to watch the video by clicking on the link above. Now in this video, we will discuss how we can interpret the phylogenetic tree obtained from the Mega 11 software. But before we dive straight into the lesson, we must understand the exact type of tree Mega 11 produces as a result of phylogenetic analysis. To understand this, we should have a knowledge of different types of phylogenetic trees and how they are classified. There are several different types of phylogenetic trees, and to understand them better, they are classified on the basis of certain criteria. We will start by these different criteria of classification. So the first criteria of classification of a tree is the presence or absence of the root. The root or the root node is the starting point of the tree. Consider a natural tree. The tree grows above the ground from the root. This means a root is the starting point of the natural tree. With the same analogy, the root node represents the ancestor organisms from which other organisms are assumed to have evolved. So if the tree has the root node, then such a tree is called as a rooted tree. And if the root node is absent, then the tree is referred to as unrooted tree. We can visualize the rooted and unrooted tree with this simple illustration. Note that the points highlighted in red are the nodes of the trees. So node is the point from which two lines arise. The lines arising from the node are called as branches of the tree. The node highlighted in green is the root node. You can clearly see that other nodes arise from the root node, and as the root node is present in this tree, we call this tree as the rooted tree. On the other hand, this is the following illustration of the unrooted tree. Here we can only see the branching pattern of the evolutionary relationships. We cannot surely say that particular node is a root node. For instance, in this tree, you can see there are two nodes from which other nodes are connected. But we cannot specifically decide that out of these two nodes, which is our root node. Thus, when there is an ambiguity in deciding the root node in a tree, then such a tree is called as an unrooted tree. We can classify tree on the basis of their topology. What do we mean by the topology of a tree? Topology of a tree is its construction or connection of nodes to each other. As we know that two different nodes in phylogenetic tree are connected to each other with the help of branches. If there is a difference in topology between two trees, then there must be a difference in the branches of the tree. Based on this difference, two types of trees are present. One is called as a cladogram and another is called as a phylogram. Let us just see how the cladogram looks like. The following illustration is of a cladogram. The lines highlighted here in green are the branches and the nodes that are highlighted are in red. The cladogram just represents the branching pattern of the evolutionary relationships among the organisms. Here, the terminal nodes of the cladogram are highlighted in yellow and represent organisms that have evolved from the common ancestor. And these nodes are highlighted in pink. As you might have guessed, the root node is present. That means the cladogram is also a rooted tree. The terminal nodes highlighted here in yellow are called as the leaves of the tree. Now coming back to the branches of the cladogram. Note that cladograms are unscaled trees. That means the branches do not represent the amount of evolutionary change or variation in DNA, RNA or protein sequence, as these sequences are used for construction of the tree. Branches represent the connection between the nodes only. So in future, if you see cladogram with variable branch lengths, then do not misinterpret it as the representation of the evolutionary change. The branch length is kept different just to make the tree look more visually clear. Now we will look at another illustration. This is the image of the phylogram. As you can observe that the only difference between the phylogram and cladogram is the variable branch length. The branch length is of considerable importance in the phylogram because they represent the evolutionary change that have taken place over the course of time to give rise to species or organisms. Longer branch lengths represent more change, while shorter branch length represents less genetic change. When we performed phylogenetic analysis in Mega 11 in the last video, we obtained a phylogram. We will bring up the same tree that we constructed in the last tutorial. Now, this was the tree we obtained in the last video. Observe carefully, the branch length is variable in this tree. Moreover, root node is also present. That means this tree is a rooted phylogram, and the branch length tells about the evolutionary change that have taken place over the period of time. Now we have understood the type of tree that is generated by the Mega 11 software. We will move on to understand the dimensions of the tree. As explained earlier, horizontal lines of the tree are termed as branches. Branches are of variable length throughout the tree. Branch length represents the amount of genetic change taken place over the period of time in the ancestral organism, leading to a speciation event. If the branch length is shorter, it implies that minimum genetic changes were required for the speciation event to occur. On the other hand, if the length is more, then more genetic changes have taken place to give rise to more evolved species. Remember from the last video that we have used molecular phylogeny to build the tree. That means molecular data to build the tree. Because we used DNA sequences to build the phylogenetic tree. Genetic or evolutionary change in this case would be the mutations in the ancestral DNA sequence accumulated over the time and eventually gave rise to new DNA sequences that we see as the genetic material of the present day organisms. Such genetic changes can be scaled using a scale bar present at the bottom of the phylogenetic tree. When such phylogenetic trees are present in the literature or scientific paper, there is always a scale at the bottom of the tree. For instance, in this figure of the hypothetical phylogenetic tree, you could see a scale bar that reads 0.007. This represents the genetic change of 0.07 or 7%. So, for instance, there is a DNA sequence of 100 nucleotides long and have a genetic change of 7%. Then there will be 7 nucleotides different from its ancestral DNA sequence. In other words, there will be 7 nucleotides mutated in the evolved organism as compared to ancestral sequence. Such a genetic change can be calculated by dividing the total number of mutations with the total length of the DNA sequence used for phylogenetic analysis. So we have 7 mutations per 100 nucleotide long DNA. Such a genetic change can be calculated by dividing the total number of mutations with the total length of DNA sequence used for phylogenetic analysis. So we have 7 mutations per 100 nucleotide long DNA sequence. Therefore, we will divide 7 by 100 to get a value of genetic change as 0.07. The vertical lines has no meaning in the phylogenetic tree. It is used for separating the horizontal branches only, thus improving the visibility of the tree. The highlighted red spot is the root of the tree. The tree has two types of nodes, namely the internal node and the second, the external node. The internal nodes in our tree are highlighted in blue, while the external nodes are represented in green. The organisms present at the external nodes are actual organisms that have been sampled, that means used as a sample. Their DNA sequences are used for constructing the phylogenetic tree. Internal nodes are putative ancestral sequences of these sampled organisms. This tree is rooted, and the root is present at the red spot or represented by the red spot, which means we know where the ultimate common ancestor of all the sampled organism was. The timescale increases from left to right. Let us consider 3 points A, B, and C on this phylogenetic tree. The point A is the internal node from which other nodes B and C have arose. Therefore, we say that ancestor A have existed prior to organisms present at the point B and C. This suggests that time runs from left to right. Near the node, you can see certain numbers. These numbers ranges from anything between 0 to 1. These numbers represent the support for the node. The number near to 1 suggests a strong evidence that nodes present on the right cluster together. What do we mean by nodes clustering together? For this, we can imagine a family tree. In the family tree, you can see two individuals, A and B. From the family tree, it is clear that A and B are brothers or sisters of each other. But there is yet another organism C. When we try to relate organism C with either organism A or B, we notice that organism C is cousin to organism A. From the evolutionary point of view, organisms A and B are more closely related to each other than to organism C. Hence, we could say that organism A and B are clustered together. Coming back to support values, greater the support values, greater is the possibility that the nodes on the right are clustering together, just like they are brothers or sisters in a family tree. The nodes that are distantly related to these nodes are like cousins or distant relatives. Hence, such the nodes or clustered nodes form their separate group called as an outgroup. Nodes that are distant relatives to outgroup are termed as an ingroup. The support values are obtained from statistical methods called as bootstrapping or Bayesian posterior probabilities. These are the names of the method that are used for obtaining the support values. Let us discuss how these methods help in calculating these values with an illustrative example.